Hi there. In this video, I'd like to go through the third part of the muscular system notes, starting on page 11. So what we've done to this point is we've really focused on a single cell, right? A muscle fiber, the different names for the organelles, how it's filled with myofibrils, how myofibrils contain sarcomeres, how the sarcomere is just this particular arrangement of thick and thin filaments and what those filaments are. We have talked about how we control a muscle cell, how we tell it to contract at the neuromuscular junction, how that signal to contract gets conveyed down the sarcolemma, falls down to the T tubules, stimulating the SR to release calcium, causing the cross-bridge cycling, right? Calcium will bind to troponin, tropomyosin moves out of the way, cross-bridge formation, and then myosin pulls actin towards the center of the sarcomere. And the net effect is every single sarcomere will shorten because the thin filaments are sliding over top of the thick. And if all the sarcomeres are shortening, that means that all the myofibrils are shortening, which means the cell is shortening. So muscle contraction is really about the muscle decreasing in length. And what we have done to this point is we've talked about it at the cellular level. So this week we're completely changing gears, and so that's why I wanted to just review that for a second and just show you here on page 11. I'm going to draw a line in the notes. Okay, so I'm going to divide the notes because up to this point we've been talking about the cell, right? From the start of the course up until here, we're talking about the cell. But this week, what we're doing is we're talking about the whole muscle. So everything that I have to say today, and at the end of the notes, it's taking the whole muscle into consideration. And so, believe it or not, the dynamics of the whole muscle are different than the dynamics of the cell. Okay, so here we're just talking about a single cell, but from here on out, today's talk and this part of the notes is about how a whole muscle works, not a cell, the whole muscle. And so what I'd like to do is run through the notes and I'll show you where I have resources for you, and I'll talk through some things that I didn't make separate videos about. So I'm going to erase my markup. Let's start with motor units. Okay, so you can see the first thing that I talk about when I talk about whole muscle is this concept of motor unit. And so motor unit, by definition, is one motor neuron. And you know how at the end of the neuron it will branch into the teledendria, and then the axon terminals will synapse with muscle cells. So that one motor neuron might synapse with, say, like 200 muscle cells. That's what you call a motor unit. So a motor unit consists of one motor neuron plus all of the muscle cells that that neuron is innervating. And so there are different sizes to motor units. So you could have a few muscle fibers, say like, one neuron connecting to 10 muscle cells. That would give you very precise control. You might see something like that, like going into the extraocular muscles, controlling movement of the eyeball, which is very precise, compared to get bigger motor units. So you could have, instead of 150 cells, 300 cells, hundreds of cells that are being controlled by one neuron. And that's going to give you more gross control, something, say, like a big muscle like quadriceps femoris. So remember that when a single muscle cell is stimulated, that it's a yes or no question, right? It's either going to contract to its full extent or not at all. Okay, so the sliding filament theory of contraction, that's a yes or no question. So that cell, you either sufficiently stimulate it, so we stimulate it to threshold and it contracts, or not. But with a whole muscle, because a muscle is made out of many, many motor units, what will happen is the muscle is going to be able to use some motor units or more motor units or all motor units, giving a graded response, what you'd call a graded response, which means that the muscle, the whole muscle, can contract with like a light contraction, so I can pick up my pen, or a stronger contraction, I can pick up my phone, or a really strong contraction, I can try to pick up my desk and maybe I can generate that force and maybe I can't. But you can see how when you look at a whole muscle, the response is graded. It's not yes or no. That's a big difference. So with a whole muscle, you can have, weak contraction or a strong contraction. And the way that that's done is through recruitment. So just to finish marking up the notes with you to that point, when a neuron delivers the nerve impulse to the muscle cell, it delivers that same message to all of the muscle cells within that motor unit. So all the cells that it's controlling will get that message. So not only does a cell contract or not contract, actually the whole motor unit will contract or not contract. When you have small motor units, like we were saying, you'll have very precise control because one neuron is controlling just a few cells compared to if you have a lot of cells in a motor unit, then you're going to get more strength, right? And not as precise fine control. So the way that we're getting the weak contraction or medium contraction or small contraction, like I said, is recruitment. So I'm going to jump over to picture. So this picture's from a different book, but I like this picture just because of the color coding and it just makes it really simple. So do you see how here we have a muscle? So we have a collection of cells, and these cells are being controlled by 3 different neurons, purple, blue, and red. And so what they're trying to show us here is that, say if just the red neuron fired, that means that, and let's say it's a sufficient stimulus, that all of the red muscle cells would contract. So not all the cells in the muscle will contract, just that motor unit. And that could give you, weak contraction. Or if, say, you weren't able to generate the force in the muscle to move the load, you could recruit another motor unit in. So the red and maybe the blue both fire, and the red and the blue fibers contract, and that would give us a more forceful contraction. Or maybe all of the neurons fire and all of the cells contract, giving us, generating the most force possible from that muscle, which may be able to move the resistance or may not be able to move the resistance. So the amount of force that's generated by a muscle contraction is proportional to the number of cross bridges formed. And so I hope that makes sense here. So if I only get the red fibers to contract, I'm going to have an X number of cross bridge formation, compared to if I get the red and the blue, I'm going to have more cross bridge formation. Red, blue, and purple, more cross bridge formation. And so the force generated by the muscle is a function of the number of cross bridges formed. And so as you increase the number of motor units on a job, the more tension, the more force the muscle can develop. And we just, obviously don't do this consciously. We just, ballpark this all the time, and sometimes we're right and sometimes we're wrong. So as an example, um, did you ever try to say like open a door and actually it was heavier than you thought? And so you try to open the door and it doesn't budge, and then you're like, oh, actually have to recruit more motor units in on that. Or maybe, you pick up an item, maybe you pick up a bottle or, pick up something off the tabletop and you think it's heavy, you think it's full, but it's not, and you overshoot that. And we generate more force. So we miscalculate that all the time, but we're actually very good at, kind of gauging the amount of force that we're going to need to generate. And so I hope that makes sense about recruitment. I wanted to talk about that because the video that I have for recruitment is someone else's video, and I just wanted to be able to talk to you about that myself. So going on to the next topic of muscle tone. Muscle tone, I understand you know what that is already before taking this class. By definition, what muscle tone is in a relaxed muscle, spontaneously will have some muscle cells in a spontaneous state of contraction. So in any typical skeletal muscle, nervous system will spontaneously activate some motor units while that muscle's at rest. So even when your muscle's completely at rest, it's not like all the motor units are just completely, on vacation. There's going to be some electrical activity causing some contraction. And that's what muscle tone is. So muscle is this constant, slightly contracted state of all muscles, and that's with the exception of an immobilized muscle. So if we have a muscle that you can't move, then you won't have tone in that muscle. And actually, you'll have the opposite of it. It will start to deteriorate, what's known as atrophy. And so the way muscle tone occurs is that we have stretch receptors in the muscle that provide sensory input. You know that. We can feel when our muscles contracted. I can feel that I'm stretching, a muscle. I don't have to look at it. I can feel that. And that sensory input is what, causes the spontaneous contraction of some motor units. And so as we increase the activity of a muscle, so if you use the muscle more, the sensitivity of those receptors increases, and it leads to an increase in muscle tone. Something else that you already know, the more that you use a muscle, the more tone that muscle will have. And I did make my own video about that, so please check that out on muscle tone. I hope that makes sense to everyone. And I'll show you a different picture as an example. So in this picture, what I'm looking at is atrophy. So atrophy would be the lack of muscle tone, and even worse, deterioration of muscle cells. And so you can see the atrophy and other issues in this picture, but this is an interesting picture too, because this person just had a cast removed. And so, I'll let you guess whether it's the black sock or the white sock of, which leg had the cast on it. I think it's pretty easy to see which limb has atrophied, and that has to do with muscle tone, the lack of muscle tone. Okay, so moving into the next segment of the notes, the whole muscle physiology experiments. This really has to do with experiments that were done leading up to our understanding of how muscle contracts. And so I think one thing that's helpful to understanding this section of the notes is understanding that, we're talking about a laboratory setting where we're going to isolate a muscle, that we deliver an electrical stimulus to that muscle so we can control, the amount of voltage, and we can also control like the frequency that we give the voltage, and we can create a myogram, which would just be like a graph of the tension developed in the muscle over time. And you can see that I share a bunch of these graphs in the notes. So I did make a kind of lengthy, for me, like 16-minute video that goes through all of these. And so I'm not going to go through all of the minutia of that. I want you to check that out so you can understand this. But let me just show you how this works generally. What you could generally do is you could put the electrode in the muscle and give it a stimulus, like a subthreshold stimulus, and nothing happens. No tension is generated. So I could do that. I could keep increasing the amount of stimulus, electrical stimulus that's given to the muscle until I reach a threshold stimulus. Then I would know what the threshold stimulus is. And when I give a threshold stimulus, this is the response that I see. I see a single muscle contraction in response to that single threshold stimulus. That's called a twitch. And a twitch has 3 phases. It has a few milliseconds where nothing happens. That's called the latent period. And what's happening there is excitation contraction coupling, right? So the message is going down to the T tubules, calcium is being released from the SR, the calcium is binding to the troponin and all that. We're not getting contraction yet. But, that only takes very short time, right? Under a second, measured in milliseconds, so before contraction starts and the force is generated. And then when the force starts going down, that's called relaxation. So you can see in our book, they color-coded those 3 periods of a muscle twitch. So they give a single threshold stimulus at time 0. The tension stays at 0 for a few milliseconds, and then contraction, and then relaxation. Okay, so that is a muscle twitch. And so what I do in the notes is I talk about increasing the frequency of this threshold stimulus. I don't in the notes go through increasing the magnitude, the voltage on the stimulus. And so we just go through one line of experiments, and that's what I would expect you to be familiar with. And so TREPA and wave summation. here you can see instead of giving a single stimulus, now I'm increasing the frequency. So what you do is you could give a threshold stimulus and I would see kind of a twitch, and I could wait till this comes right down to 0 and then give it this a second stimulus. That's called trepa, when you let the tension reduce down to 0 before giving it the second stimulus. But here you can see we dialed up the frequency. And so we're halfway through the relaxation period, hitting it with a threshold stimulus. So the stimulus is not greater, it's the same. And you can see that the muscle responds. And again, it responds. Okay, and so on. But you can imagine if we keep increasing the frequency, you're not going to be able to keep getting greater and greater tension, like we can see here. Too bad. We could be like superstars at the gym, right? Every time we contract the muscle, it gets stronger and stronger. So we're going to, plateau. And if you plateau in this way, this is like a real life example. This is what's called incomplete tetanus. This is basically how we make our movements, that when the nervous system fires, that it fires these, stimuli at a certain frequency so that when our muscle contracts, it stays contracted. That's what allows us to stand, right? To sit, to write, to do the things that we do. We're not operating in individual contraction and then a full relaxation. But in the lab, you can see complete tetanus, which is, plateauing in a smooth, sustained line. This is a little, this is unrealistic for every day, but in a lab, you could generate this by giving the stimulus so frequently that the muscle contracts, stays contracted at its full tension, and then when we remove the stimulus, that the muscle relaxes. So I do go over that all in this whole muscle contraction video. And just to take a look at our playlist, this is the video that I'm talking about. So whole muscle contraction. And I use a picture in the book and I go through all of those graphs, and I take my time doing that. So please watch this, and that will give you some good background on what we're talking about. So, and I also talk about isotonic and isometric contraction in that video, which you might be familiar with already. And just in case, you didn't write this down, I mentioned this earlier in the video, that the force generated by a muscle is directly proportional to the number of cross bridges formed. So I wrote that down for us too. So then I'm changing gears a little bit, and we're talking about, okay, so we understand how the whole muscle functions. How do we get the energy that we need for the muscle to function? So muscle metabolism really is just, like what processes are we going to use that's going to generate the amount of ATP that we're going to need for this cell to function. Okay, so ATP is the only source of energy that's used directly for muscle contraction. So why do we need all of this ATP? So I ask you the question, what cellular activities in the muscle, require ATP? And some things that come to mind right away are, like think of the sarcoplasmic reticulum. It's using active transport 24/7 to keep calcium within the cell. During contraction, when do we need ATP? Think about that. We need ATP for the detachment of the cross bridge, right? If I ever ran out of ATP, I'd be stuck in a contracture or a cramp. So there's two good examples, one at rest, the sarcoplasmic reticulum, and one during action, during contraction, the cross bridge detachment, of when we need ATP. So we need a lot of ATP for muscle to function properly 24/7, whether the muscle's at rest or whether it's contracting. And so, how is ATP generated? ATP is generated by, do you remember the organelle? You probably do. It's generated by mitochondria, and mitochondria can't just, make ATP out of the blue. The initial ingredients necessary to make ATP, mitochondria require two things, and there's no way around this. You need a nutrient source. That nutrient source could be carbohydrate, protein, or fat. Okay, so mitochondria can use any of those macronutrients to make ATP. Typically in a bio class, we talk about glucose, sugar, right? A carbohydrate being the nutrient that's used, which is fine. But we could also use fatty acids and amino acids to make ATP. And then the other thing that you need, so we need a nutrient source, and the other thing that we need is oxygen. Mitochondria rely on oxygen. Oxygen is the final acceptor of electrons. If you're familiar with that, I talk about that in the video that I made. So I'll let you watch that and get that, or maybe you're familiar. But those are the two things. You need a nutrient source and oxygen. And so here's what I'm thinking. This is if I didn't know any better. Can the ATP be produced and stored up for later? So like, in other words, when we're at rest, could the mitochondria like make ATP, stockpile the ATP, save it for later, and then when my muscle contracts, I can just use that? That would be what I would, plan on. But unfortunately, ATP cannot be stored for later. Instead, there is kind of like a variation of storing it for later. The closest we can get to storing it for later is using this molecule creatine phosphate. So creatine phosphate is basically, a molecule that you can add a phosphate to. So we could take this creatine and you could add the phosphate from ATP to the creatine, making creatine phosphate. And of course, that would leave us adenosine diphosphate, ADP. But here's the thing, when your muscle starts to contract, because I can make a lot of this creatine phosphate and stockpile it and store it up for later, then I could quickly run this reaction in reverse, take the phosphate off of creatine, drop it onto the ADP, giving me ATP and creatine. So I did make a video that goes through this in a more, leisurely way. And so please watch that. So I talk about the use of creatine for muscle. And when our muscle starts to contract, the way that I wrote my notes, which is different from the book, is I'm writing my notes in the sequence that the muscle would use the energy source. So the first thing we're going to deplete is the creatine phosphate. We're going to use that first. After I use the creatine phosphate, then I have to use aerobic respiration. And what the aerobic part means is that it requires oxygen, right? Because basically what we're talking about here is that the mitochondria is going to generate the ATP that the cell needs to keep working live time. So the mitochondria have to keep up with the ATP demands, whatever your activity is, running the marathon, lifting weight, live time. And of course, there's a question mark there of, can that happen? So the mitochondria are going to work as hard as they can to keep us provided with ATP. And if at any point the mitochondria come into a relative deficit, okay, so here, mitochondria has a relative deficit of oxygen. Why would that happen? We're still breathing, yeah, but because oxygen is needed by all of our whole body, right, not just the muscle, we might not be able to give the muscle all of the oxygen that it needs to keep going with aerobic respiration. And when you have these relative deficits, what happens is we can't use mitochondria to generate ATP. So now we're on to plan C, right? Now we're on to the third thing, which is anaerobic respiration. And anaerobic respiration, believe it or not, is just the initial version of what we already talked about. So I talk about this in the video. Basically, what we do to move glucose into the mitochondria, which is a process called glycolysis. It occurs in the cytoplasm, generates, a little bit of ATP, 2 molecules of ATP per glucose. That's what we use. And then, we generate pyruvic acid. Typically, we move that pyruvic acid into the mitochondria, as I talk about in the detailed video. And we convert this pyruvic acid, we extract, electrons from it to generate a bunch more ATP. But because you can't move this into the mitochondria, what happens is it stays in the cytoplasm, and we have enzymes in the cytoplasm that will convert the pyruvic acid into lactic acid. And so we start making lactic acid as a waste. And then we can just carry that away, and the liver can actually take that lactic acid and unbelievably, the extremes that we go through to keep ourselves going, the liver has the enzymes necessary to drive glycolysis in reverse. So the liver can actually take the lactic acid, convert it back to pyruvic acid, and then convert it back to glucose. That's called the Cori cycle, and then the liver can put that glucose back into the blood to fuel more work. So, it's incredible. So I did make a video about aerobic and anaerobic respiration. Please watch that because that's where I'm going to go over really like the details of what I'm talking about in this section of the notes. Which brings us, to the end of the notes. So a few final things. Fatigue in muscle is due to the relative lack of ATP. Like I said, if you have a relative shortage of ATP, you get stuck in a contracted state because we need the ATP for the cross bridge detachment. But it's not like a real lack of ATP. That probably really rarely happens. There's also damage due to the SR, damage due to the cell, so you get ionic imbalances, and all of these things kind of add up to fatigue the muscle. And so once we stop working, what we'll do is we just run everything basically in reverse. So we convert the lactic acid in the cytoplasm of the muscle cell. So you can do this right in the muscle cell. You can convert that back to pyruvic acid and then put that pyruvic acid, oops, sorry about that. Okay, we can put that pyruvic acid back into the mitochondria to undergo aerobic respiration so we can make ATP. We can turn the lactic acid that's still in the cytoplasm of the muscle cell we can turn it into ATP. We can also, when we start making this ATP, start taking the phosphate off of that ATP to start stockpiling some creatine phosphate. Glucose that's coming into the cell, we could store it up as glycogen, right? So we could store glucose to use for later. And in muscle, don't forget, we have that molecule called myoglobin, which can bind to oxygen. And so we'd even be able to store some of the oxygen. So muscle has the ability not only to store some energy in the form of creatine phosphate, but to even store some of the fuel we'll need, store some glycogen and some oxygen. So that's what we'll do during recovery. So depending on how we use our muscles, we will start to train our muscles for our activities, which is, like part of physical training. So if you train to run a marathon, like an endurance type of sport, that's very different from, say, like weightlifting. And so the variance in how we use our muscles will start to train the muscles. The muscles are actually going to start to acclimate to the type of activity the person participates in. And so we have these two categories of muscle cells or muscle fibers, right? The same thing. And there's more than two. These are subcategorized, and I did put a video in my playlist about that, which is more optional. I'm really concerned that you know these two categories into fast fibers or slow fibers. And so I'm going to look at a picture for this and see if this makes sense. And so fast fibers, these are fast fibers that you can see here. So they're looking at an extraocular eye muscle, a muscle that moves the eyeball. And do you notice how like big these muscle cells are? So what we're seeing with fast fibers is they're strong. They're packed with myofibrils, right? And typically with muscle hypertrophy, that's the way we do it, that the number of cells stay the same, but the number of myofibrils within the cell increases, and so the diameter of the cell increases. These cells have good glycogen stores, and because you get a lot of cross-bridge formation, right, they'd have a lot of force. And so you can see these are called the fast fibers, or because they have a lower amount of myoglobin, they have a lighter color. People call them white fibers. So white fibers or fast fibers are large diameter, paler color, easier fatigued, right? Because we don't have the storage and of oxygen and the rich blood supply going to the white fibers. They're really kind of suited for like a fast activity. So if you're a sprinter, you're going to develop this type of fiber compared to if you're a long distance runner. Long distance, that's going to be more endurance. That's a red fiber or a slow twitch fiber. And so here they're looking at at cells from the gastroc. So we walk, so we would expect these fibers to really have a lot of endurance. And when you look at these under the microscope, they're darker in color. They have a rich blood supply, lots of myoglobin. Okay, so and myoglobin is a pigment just like hemoglobin. And so they're red, and people call these red fibers. You can see they're smaller fibers, so they're not as strong, but because they have so much mitochondria, blood supply, they're fatigue resistant. So they're not as strong, but they're very good for endurance. And so any given muscle of ours will have both types of fibers. They'll have red and white fibers. It's just that when you look at a muscle, depending on your activity, you can train it to be predominantly one type or another. So that's what this is about. And then hypertrophy. Hypertrophy, I already mentioned. Hypertrophy is when you increase muscle mass due to increased physical activity. And the way that that happens is increasing the number of myofibrils and increasing the glycogen storage. Of course, mitochondria will increase, but it depends on, usually like when people hypertrophy a muscle, it's a white fiber, and so they're really not going to have, like lots and lots and lots of mitochondria. And so to go back to the slides as an example, with muscle hypertrophy, this would be an enlarged muscle size, not because the muscle has more cells, but because each of those cells is bigger. It has more myofibrils. So taking a look at our video playlist one last time, here's the video that I made about creatine phosphate. I go through, in my face-to-face class, I go through the details of aerobic and anaerobic respiration. We spend some time on that and go through the details, so please watch that. That's where I do that. And then here are the more optional videos. So this you might be interested in, fast twitch versus slow twitch. Same thing here, and here they show you how the fast and slow are sub-categorized. I have a video on muscle fatigue. That would be optional. And then hypertrophy. I thought you might be interested in those. But again, I'm not going to have test questions just on that video, the subcategorization or any of this. So I hope that helps, and I'm looking forward to hearing your questions. Thank you.